Good morning, everybody. This morning session we start in English. And the reason why is that we have Professor David Thomas here today from Australia. He is really a gem for us to have, because not only is he an excellent scientist, but he also takes a lot of energy and pride in organizing his institute. He is a director of Kinghorn Cancer Center and head of the cancer division of Caravan Institute in New South Wales. And actually to have this ability to organize cancer research and cancer treatment in a very elegant way, that is also what we are looking forward to hear from you today. So after we have had the opportunity to present him formally, we will also say that probably the reason for that we managed to have him here is that this little monster to the right is actually David Thomas. He has become a keen Norwegian friend, so therefore we hope to see you back later as well. So we hope that you now will take the floor and give us some of your ideas and your experience. Welcome.
Thank you very much. It really is a pleasure for me to come back here. I am extremely fond of Norway. It strikes me as being just a colder version of Australia. Large landmass, small population, many of the same challenges. We depend equally upon our natural resources for the wealth of our community, and we are faced with the challenges of how to integrate genomics into our medical care. So what I'm being asked to talk to you about today is about what I believe is the next wave in genomics and medicine. I'm sure it's been reflected in this meeting's discussions, but it's generally an issue broadly. The first wave, one might say, of genomics in cancer biology is about cancer biology and its drivers, and that's been going for about a decade now. And on the wave of that information, there have been many direct-to-consumer marketing efforts in translating these technologies into the marketplace, but it's always been dependent on the assumption from the patient's perspective that the test, the genetic result, is somehow the same thing as a treatment, which as clinicians we know is not the case. And I think this second phase of genomics and cancer medicine will be about connecting those two parts together so that patient expectations match with what we're trying to do. And I should say also that in my view that this necessarily entails an integration of research into clinical practice, which of course is a very great challenge because medicine is an inherently conservative and relatively young discipline. It's designed itself around risk aversion, first doing no harm and so forth. And yet we're in an era where I think the opportunities to benefit patients are occurring so fast that our health system, the way in which our health system is constructed, becomes one of the barriers. And that's one of the themes of the talk I'd like to present to you. And yet at the same time, I'm not suggesting that we suddenly change cancer care. The reality is, so great is the impact that governments are going to have to spend probably billions of dollars in each community of any size in order to implement the changes ultimately. And I don't think we have the evidence base to justify those sorts of investments in a health economic sense. So I think for this next little while, what we'll have is this virtuous cycle between clinical practice, clinical research, and basic research where the information gets fed forward live. And that sounds like a simple thing to say, but it has very practical implications for how we conduct those clinical trials, who takes part in them, and where those trials are conducted. And I'll try to indicate some of what my thinking has led me to propose as a model in Australia. And I think many of these implications of this model will apply here too. So the fundamental premise here is that cancer is a genetic disease. I don't need to labour this point, except to say that despite the fact that immunotherapy has become a very important part of the future of therapy of advanced cancers, and maybe certainly including adjuvant treatment of cancers aiming at cure. Nonetheless, cancer is fundamentally a genetic disease. I suspect over the next few years You'll see the marrying of immunotherapy approaches to cancer with genomics, because it will become an important question as to why some people respond and some people do not. And the specificity of immunotherapies will in turn depend upon how cancer cells genetically evade the immune system, a question which simply hasn't been answered yet. I think the immune system is just a very important example of an environmental selection pressure for the environments in which cancers occur. The second fact, which is very important and sets the stage here, is this extraordinary generation of knowledge. If oncology is roughly 40 years old, and that's all it is, two human generations old, this has occurred in the past 10 years. And it's important to reflect upon this, the shape of this curve, because it's essentially presents one of the major challenges. It is not possible now to use a clinical environment in which progression is like this when we have such dramatic... Okay. Thank you. So this is a really important concept that needs to be borne in mind. The reality is not only this exponential increase in knowledge generation is an important issue, but the fact is, through the internet and communications technologies, the discoveries here are being introduced into the community and raising expectations faster than we can handle it, creating enormous pressure on regulators and healthcare professionals, administrators of institutions as to how to integrate these things. And this is not a problem that's unique to any one country. This is something from 2010, so I thought I would show you the current state of play, which is that currently we can generate 0.6, actually close to a terabyte of data per day in 2014. That was last year, and that will be going up. So phenomenal increase in capacity and depth of understanding cancer. But the other thing that's happened is this cost reduction, so that in January 15 of last year, almost 14 months ago, the cost of sequencing the whole human genome has essentially dropped even faster than Moore's law would predict. And again, it's this acceleration which is the major concept I want to get across to you. It's not just that we understand more, but we can now start to democratize this. This means we can apply it not just to rare cancers to understand, pretty patterns. That's the nature of technology and science, is that the first papers come out describing a handful of samples, typically done in a major center which has access to the technology. But very rapidly, this goes out into the population as a whole. And that's important because, in the end, health systems care about not the exception, but actually the broader community. But this technology is effectively entering into the clinical space. What does this mean, this $1,000 US per genome? Well, in Australia, I did a brief survey of 23 clinical cohorts, disease cohorts, comprising over $200 million of investment in annotating various disease entities, many of which have a genetic basis, and comprising over 200,000 individuals who had been subjected to a technology over the past 10 years, genome-wide association studies, which cost about this when they were first introduced. So there is no doubt that this technology will be expanded out and applied very broadly over the next decade. And from a cancer point of view, these anecdotes about using the information then to apply treatments to patients with these spectacular results, which patients are aware of, the improvements in survival, the challenges of the fact that we don't cure people, all these things start to become clinical issues which are important for cancer care. And this model, this is not a new slide, this is 4 years old now, from Laura McConnell. And this model about the principles of personalized or precision medicine have been around for some time. The question is, how do we translate that into practice? And that's the question that people face. So I mention this because I want to put into context global investments. Genome England, this is courtesy of Mark Caulfield, who's the Chief Scientific Officer of Genome England, have invested £100 million, that's I think probably of the order of £1 billion kroner, into sequencing 100,000 whole human genome sequences. But their principle is not now discovery. Their concerns are generating health and wealth from this investment. This has come from David Cameron directly. He made the decision to invest in this space in part because of his personal experience with one of his children, but partly because as a businessman, he's thinking about where the wealth is going to come from in the future. So the question is not about discovery anymore, but how you apply this to the population and how you impact. And there are 3 broad areas that Genome England have identified. And one of them is developmental disorders. I'm not going to talk about that more, other than to say that I think the first and most immediate application of genomic technologies in the clinical space will be to compete with existing technologies that are applied for developmental disorders, intellectual disability, various forms of inherited or congenital and currently unexplained illnesses in children particularly. But the second is in cancer, and the third in this, which I won't again talk about, is in pathobiology and the genetics of pathogens within in Genome England. So in cancer, they're going to test this principle. They're going to look at questions of tumor heterogeneity, evolution, but also of stratified medicine. And they're going to focus particularly on a subset of cancers, but including rare cancers, something that I think is important. I'll come back to later. And they've got a program which they're developing, particularly in stratified medicine, particularly focused on lung cancer, where they're trying to test models of interventions. So it's an important principle that Genome England, the investment of £100 million, is not for the investors about genomics and discovery. Primarily, it's about how you turn this information into healthcare. So, as I said before, I don't think one can go straight from genomics into simply changing the practice of medicine. That's not the way that medicine works. It's done through clinical trials and the generation of evidence. But it's important to consider what the state of that is at the moment. So I'm a believer in classical clinical trials design. I believe that we will not supplant this model of going from phase 1, looking for toxicities and how we optimally use new drugs, through phase 2, getting an estimate of the activity of a particular agent in a particular situation, and then into phase 3 where we compare it to standard of care. I think that's going to necessarily be the case. But these study design, which has been around and tested and contributed to improved outcomes, has been around for 30, 40 years now. And it developed at a time when we had low-cost therapies, we had no idea of how the drugs worked, we had low response rates overall. Even if the population as a whole had a benefit, it was in typically a smaller fraction of the population. It was applied to large populations as a consequence because we couldn't select those cases. We did it in breast cancer, and we treated 4,000 or 5,000 patients on an arm to show that you have a 5 to 10% improvement in survival. And that's considered statistically meaningful, but it's perhaps not the model that we use today. And the development times are typically slow in the sense that the pace of knowledge generation is not that exponential increase that you've seen today when these things were developed. Toxicities were severe, and there was a sort of environment of therapeutic pessimism in the community, but also in our colleagues who work in the clinical space. But that's changed, as you can sense. We have high-cost therapies, so these things are important because if you have to pay for a drug off a trial and it costs $100,000 per patient, the question is how much should a community invest in conducting a clinical trial? It might not be worth it if the drug cost is $500 a year as opposed to $100,000, but we haven't considered whether these models of how the system invests in clinical trials as effectively a standard of care might now be different on the basis of the cost of those therapies off trial. Rational design is important. We have these targets. When we design the trials around those targets, we start to put things together in a much more intellectual way. In fact, I would argue that we must not lose sight of the fact that human intelligence is overrated as a mechanism for discovering opportunities, and we must still use our empirical and clinical observation to design our trials, because there are things that we don't understand. RAF mutations predict response in melanoma, but they don't predict response in colorectal cancer. There may be other situations where drugs are tried. For example, CDK4 inhibitors in the context of some tumors may not have any effect, but it does not mean that it will not have effect if we just simply explore the remaining opportunities. And the question is, how do we combine both intelligent, rational design with empirical exploration in the clinical space, taking advantage of something which I think is hard to codify, which is clinical intuition, which I think is extraordinarily valuable in the way, and something that we should think about trying to harness. Of course, the populations are going to be smaller. You're going to be looking at a subset of breast cancer, a subset of something that's marked out by a particular mutation. The development times, as I said before, are increasing. The toxicities in principle are lower, but that's not universally true. And there is an enormous degree of therapeutic optimism and community awareness, which I think is an incredibly important driver that we need, that ultimately determines why governments should invest in this space. It's because governments represent the people, and the people actually want this because cancer is a major blight on the community. So, how well are we doing with that model? Phase 1, 2, 3. Well, this review article is well worth looking at. This side of drug development, preclinical side, has extraordinarily expanded over the past 50 years. It follows the exponential changes. But the clinical trials component, which has been relatively sluggish, has not advanced at the same rate. This is a comparison between how we used to conduct trials back in the 1980s and 1970s and 2010. Back then, the protocol was 10 pages. The protocol development took weeks to implement. The cost per patient was under $3,000 per head enrolled. The time from submission to opening was 2 weeks, and the consent form was 3 pages. It's now 150 pages. Protocol development lead time takes years. The time from submission to opening is average of 20 months. There are 370 pre-activation steps. The consent form may reach 30 pages, leaving outside the companion biomarker studies we have to consent to. And the average cost per patient, as of 2 or 3 years ago, was $120,000 plus for the average phase 2 or phase 3 participant. That meant that the average phase 2 study, as of a couple of years ago, cost $10 million and takes about 5 years to complete. And the average phase 3 study costs $40 million. And the reality is, currently, using our system, half of these fail, and then of those that go to phase 3, half of those fail to proceed, and of those, a subset actually get approved by regulators. And all of that drives a situation where, in effect, the exponential increase in our capacity to know things is ironically being contrasted with an exponential decrease in the ability of us to translate that into clinical trials. And that's in our space. That's not a scientific question. That's a health systems question. That's a clinical medicine question. That's an administrator's question. So the question is, can we think up ways to address this particular barrier if we are to achieve the object of trying to translate genetics into things that will help patients in front of us? The consequence of it is, though, that the number of drugs invented per $1 billion has effectively exponentially halved every 9 years for the last half century. This just proves my point. The fact of the matter is that that increase in ability to discover has been materially associated with a decrease in the efficiency of conducting clinical trials. It also creates an irony. So if we were to succeed with every one of our brilliant ideas, could our communities afford our success? I suspect that that question is something that we need to think about earlier rather than later. We can't simply solve the technical problems and create scientific and patient-driven outcomes without considering what the magnitude of the effect is on the system as a whole. And I think it's going to call for a new model of partnerships to try and reduce the overall costs to our communities of conducting of clinical care as well as conducting clinical research. And this is not something I'm making up. These papers, especially those by Hagop Kantarjan and David Steensma, you can see here, are well worth reading. They go into some of the extraordinary changes that these experienced clinicians, particularly Hagop Kantarjan, who is a giant in the field of hematological malignancies, has seen the past 30 years with of clinical research. He can see what's changing, and I think these articles which have come out in the past 12 months or so really drive the impression that we are entering a crisis. So, but of course, this is not something new. There are clinical trials. So what type of clinical trials attempts have there been? Well, broadly, there are this basket trial and umbrella trials. So the basket trial, I think of the most beautiful trial design that I've come across for a long time was the B2225 study, which was when imatinib mesylate, Gleevec, was first introduced. Novartis introduced this trial after the success in GIST and CML, looking for opportunities where this drug might work. And they had a trial design which had many potential indications. And a clinician could come along and say, look, I've got a patient, I'd like to treat them on the B2225, and here is my rationale. And they put a submission into an electronic communication eligibility committee who would then approve it, and then the patients got aboard. This particular trial identified an opportunity in 8 patients with a disease called dermatofibrosarcoma protuberans. A colleague of mine, Grant MacArthur, was the lead author on a paper that came out in JCO in 2005 from this study, because dermatofibrosarcoma protuberans has a translocation involving a target of imatinib PDGF receptor. It's a beautiful clinical trial design, and it's certainly an important part of a future of conducting clinical trials to look for opportunities to develop drugs. The second type of trial is where you have a screening population, an umbrella trial, and you might select it to a particular population, like the BATTLES trial, which I'll come to in a minute, but you have many different drugs attached to the broad pipe of genomics. So the whole point of genomics when you do these tests is you do 20, 50 tests at once, and then you presumably have 20 or 50 interventions that you can offer at the end of it all, which starts to create some very challenging numbers when you think about how to conduct a trial of that kind, how to afford to run a trial. Can you run 50 parallel phase 2 studies at $10 million each, and each taking 5 years? Is that possible? Well, people have been thinking about this, but they narrow it down to a particular disease. So in the case of the BATTLE trial in lung cancer, you have a patient with lung cancer who gets a biopsy. There's a limited set of tests which is undertaken. You can see here it's for KRAS and BRAF and VEGF receptor expression, when it was thought that those things predicted response to angiogenesis inhibitors. And then a group of 4 different treatments, and effectively in a trial. And here you have 341 patients registered. And this is an important point. Not everybody who goes onto these studies will be able to go onto the trial. 86 patients did not get onto the trial, essentially because the patient's trajectory was deteriorating faster than it was possible to do the molecular testing and then feed the patient information back. I think this is important. For example, right now there's a study which is ongoing in Australia for pancreatic cancer. Pancreatic cancer is a disaster to be treating in this particular way, because the median survival and the state of patients from the time where you get a biopsy, which can be difficult in a retroperitoneal organ like the pancreas, and the time when you might have to intervene, particularly with opportunities between the diagnosis, like Abraxane, opportunities that patients may take, has meant that over 2 years there has been no patient who has been subject to a screen who has actually been eligible for targeted therapy. I think we need to understand that there is a really important set of drivers about the way in which we do these molecular tests and have interventions that may be important to actually making these sorts of challenges real. Patients don't often have the luxury of being able to accommodate some of our current paradigms for genetic testing in the clinical space. Of the 255 that were randomized, 97 were allocated randomly to these various treatments, and there's a Bayesian design, which I won't go into. This I show because it gives you a sense about what you might expect to see. So here you have people with mutations in EGFR, and they have a 35% response to erlotinib, but not actually much different response to a variety of other agents. The standout here is sorafenib for people with overexpression of VEGF receptor, sorafenib having some activity in those diseases. But overall, it's not black and white. And you can see that in this study, there were 58 patients treated, 52, 36, 98, at roughly $120,000 per patient enrolled. This is an extremely expensive study, which I don't think has shed a lot of light, perhaps because of the nature in which we do this molecular testing. But this is an example of a trial that was initially set about. This is a different trial, again focused on a particular disease, in this case breast cancer, published in Lancet Oncology. Again, 423 patients enroll, 407 had biopsy results, 104 were excluded because of problems with the biopsy technique itself, low percentage of tumor cells or no frozen material. Only 299 out of, or about 3/4 of the patients had DNA suitable for analysis, and then there were other set of exclusions. Ultimately, only 55 patients got treated, which is a really interesting problem. And part of this is about the technologies that were used in this particular trial, which are evolving. But it goes to show that when we're conducting clinical trials, especially where there's so much expectation and so much cost, it is important for us as clinicians to think about where to conduct these trials so we have wins. And that isn't, I don't think the way in which these initial studies have been conducted are particularly designed to be successful. You can see that the rate of response is 3 of 6. Here's the other extreme. You know, here we have treatments in 6 patients. And again, I don't think 6 patients is enough. I think 58 is too many, and I think 6 is not enough, which gives me a number of roughly between 10 and 13 patients, which I'll come back to in a little while. This is probably the most interesting study that I've seen. It's conducted by the NCI. It's called NCI Match. And they're intending to enroll 3,000 patients with advanced disease, genetically sequence them, and then put them onto a range of essentially phase 2 studies. That's because the NCI can afford to do this sort of thing. You can see classical phase 2 endpoints, overall response rate, progression-free survival. There is something of a signal-seeking design here, Simon two-stage design of 30 patients in total. And something that is very important to me is this down here. They're specifically trying to address some of the inequities that are beginning to emerge with studies which are focused upon the common cancer types. In fact, I would think that, I will come back to this in a moment, but I think one of the unexplored opportunities is how to deal with the neglected and orphan diseases, which comprise the single largest burden of disease in cancer in our communities. I'll come back to that in a minute. But I think this attempts to do that. One of the important components is that the NCI has extraordinary pull with pharma. This has got to be, in my view, a partnership with pharma. Pharma should see this as a way of using community investment to screen populations with expensive tests, which are in an era where that's going to be increasingly either borne by pharma directly or it's going to be borne by the community. And I think the NCI has got a lot of companies who are prepared to put their drug pipelines. And here are the targets that they've screening for. And here's an important aspect actually to the study which I think is important in designing trials in the future. Because knowledge is being generated so quickly, I don't think it's possible to write a protocol in the old-fashioned way where you specify a whole series of decision forks, anticipating all potential outcomes. And I think that's because when you design the trial within 2 years, that information may change. There are going to be variants that have not previously been identified. There'll be papers coming out in PNAS showing that activity in certain disease subtypes. There'll be studies coming out which show that that drug does not work in that particular combination. I think it's going to be increasingly important for trials, especially in populations with advanced cancer with unmet needs, to have a live capacity to review data at the point where a clinical decision needs to be made. That's not something that's essentially a bit like the independent data monitoring and safety committees that we have in many trials in the phase 1, 2, 3 model, but actually involved also in the adjudication about eligibility and so forth. I'll come back to that, but I think it's really important that we design our protocols from an ethical point of view so that there is rigor and safety and protection for our patients, but at the same time allows latitude over a study which may take an event where the event horizon is going to be longer than the generation of new knowledge which may be relevant to interpretation or execution of the trial. I was talking about rare cancers. I think this is a really important principle. This 1,000-year-old definition of cancer was broken down from 1850 onwards into a model system based around the light microscope, and now we're in an era of molecular pathology. And what that means is there are several really important things for clinical trials design. The first is, as I said before, this is not one disease, but in fact probably 20 diseases. And that's going to be relevant to the choice of the drugs that we consider. But it also means that some rare diseases, which would not have been considered routes to registration for drug, for example, now actually share a whole lot of potential targets with diseases that are common. So in principle, one of the potential opportunities, the promises of a molecular era of medicine is that the rare and neglected cancer population may actually, for the first time, start to become tractable. You start to get something that makes everybody a rare disease. The idea of personalized medicine is all based around the idea that the mutations you have in a particular cancer may be unique to that individual and so should treatment. So why is that important? So this is data from Australia which shows for males and females the incidence of new cases of these cancer types, and then the number of deaths, the percentage of the total number of deaths, the rate of deaths, and importantly, the person years of life lost to those diseases. So what you see is, of course, prostate cancer. These numbers are very high. Breast cancer is very common, and there are some other diseases which are less common. The other group, which are the less common tail of this list, actually is almost the single. It's the second most common in males, and it's in fact also the second most common in females, but in terms of years of life lost, is actually far greater than any of the other groups. And this group has done particularly badly because it hasn't been tractable for old-fashioned clinical trials design. You can't do a phase 3 study of 4,000 sarcoma patients, even if it were one disease, which it isn't. It's just not possible. So these things, what that means is the principles that clinical trials are the best standard of care has translated into this excessive lethality for the understudied group of cancers. From a community perspective, battle-type trials will study lung cancer, and breast cancer trials like Saphira-1 will study breast cancer. I think we have an opportunity, and I'm speaking now because I treat patients with a rare sarcomas, rare group of cancers, to use this new era of molecular medicine to try and ask questions about how we can impact upon this particularly large fraction of the morbidity and mortality due to cancer. And so I think that should be an important thing that we should think about in terms of our clinical trial designs. And in part because every year there are more and more therapies that are becoming available to people with rare diseases. And I think this is likely to, they project by 2020 there will be 200 new therapies available for rare diseases. The opportunities for intervention are increasing. These are not, this is not a situation where we can make a diagnosis but we can't develop a treatment. The treatments are on one side of the equation for these rare diseases. The treatments, the diagnostic capacity is in the capacity of genomics. So what all these factors have led me to consider is whether or not there may be an opportunity that inserts itself, doesn't replace, but inserts itself between phase 1 and phase 2. When I reflect on my clinical practice in sarcoma, there's often a particular close relationship between a phase 1 unit and the sarcoma unit. And the reason is because we can never find a trial which is designed for a sarcoma patient. So we see these new drugs coming through and we think as clinicians, a CDK4 inhibitor in phase 1, that's how I'll get my sarcoma patient onto the CDK4 inhibitor study. But the reality is patients don't want to go on to a toxicity, a phase 1 trial. From their perspective, they're after treatments. So is it possible that one could design a trial which is which is driven by molecular criteria, which is not as expensive or as formal as a phase 2, but it is interested not in toxicity but in responses? Could that be useful? And so what I thought was, we have a model for that in the Simon two-stage design. In the Simon two-stage phase 2 study, you have an initial set of patients where you look for a threshold of activity, and if you don't see it, you cut the trial, saving lots of money and avoiding treating unnecessarily patients when the outcomes are foregone. So what happens if you just looked at that Simon first stage? You did this for pharma. You didn't expect pharma to do the screening. You didn't expect pharma to invest in this. That was something that the community was going to invest in. Would pharma be interested? Well, if pharma has 1 in 2 phase 2 studies fail at $10 million a pop and 5 years of investment, and you could identify, simply because you were engaged with pharma in seeking essentially lots of parallel Simon phases of design, that would give them a broader range of opportunities to develop their drugs from a regulatory perspective and invest their money in such a way that their probabilities of success would be greater all the way through. This is the concept. So what we propose is what we're calling a genomic cancer medicine program, and this is the molecular screening and molecular therapeutics component. We're going to screen 1,000 people in Sydney for various techniques, not just genomics, because I don't think it's all about genomics and genetics. Some of it's about immunohistochemistry. Now, patients will have potentially actionable mutations, and there will be others who don't have any actionable mutations. And that's an important group, which I think we need to think about, in part because the immunotherapies may be a perfect treatment arm for this group here for whom genetic-driven therapies may not be available. It depends upon what those numbers are, but I can tell you at the moment they look like they're 75% of the screen population. So only about 25% will go into this group. And this group here has, goes through a molecular tumor board, some sort of evaluation process about whether the mutation is reasonable, and then patients will either be enrolled onto the study that I'm going to describe to you, or they're referred back to their clinician for a standard therapy, which may exist in some cases, or there may be another trial which is dependent upon molecular screening. So there are multiple potential points of intervention, and this is the one that I'm principally concerned about. So if the patient has a mutation for which there is not an existing therapy or a trial, we've designed a framework down here which is designed to offer lots of opportunities in parallel, commensurate with the bore of the screening process. So we need to do this, we need to have at least 10 or 12 different treatment options, and at the same time this has to be affordable. The budget we have for this is roughly 3.5 million Australian dollars, not the 100 million pounds. So this is intended to be an exploratory exercise. So one important component of this module, of this concept, is that we have a framework or a parent protocol. The idea here is that you go to an institution which treats lots of cancer patients and which has an active clinical research program, and you go to the ethics committee and you say, here we have a population of patients who have unmet needs and we don't have the capacity to have a trial for each one of these patients. So what we want to do is set a framework for a series of rationally designed clinical studies which are intended to be small, fast, and ask questions very directly, competing, in my view, not with phase 1 or phase 2, but actually with the PDX programs that many of us use to provide evidence for a concept in the lab. So in a lab, if you come from a lab, you might have a great idea for a treatment of a patient, but because $10 million to do a phase 2 study and it takes you 5 years, the barriers to actually translation are very high. So what you do is you go and you get some patient-derived xenografts, you treat mice over a 2-year period, and you show that some of those mice respond, and it goes into the last figure in a cancer research paper. So the concept here is, instead, you go to the clinicians at your institution. You spend 3 months filling out a module which is within an established, ethically approved framework, where you cite your evidence. You show that the patient population has no alternatives and that they will benefit from at least asking an intelligent question. And that has to happen fast for this to be worthwhile. And then you get the researcher to give their biological rationale as part of the premise. The threshold for the evidence required for the intervention is lower because you're doing it at an institution in a population with unmet need, and you're driving it on research principles. And within 6 to 12 months, hopefully you would have some idea about whether that concept which the researcher has some value for the patient population. And you do it more quickly than you would do otherwise, and you do it more broadly than you might do otherwise, creating a richer series of opportunities for then going back to the phase 2 process. So this protocol basically may take a while to establish, but it's a sort of an intellectual framework to be conducted within a clinical trials unit for a series of modules which are driven by science. And that's what a child protocol is. I'll come back to how we design a child protocol in a minute. The child protocol is designed to go back to the 1970s model of clinical trials. It's meant to be a 30-page protocol. It's meant to be a 2-page, 3-page consent process. It's meant to be a small number of patients, and it's meant to be geared to sensitive, clinically relevant assay endpoints rather than formal phase 2 endpoints. And because it's light, it should be relatively low cost, and we should be able to answer these questions much more quickly. So here's the concept. You screen the patient population and you go into one of these modules. You might have 10 to 13 patients, typically the size you see in the Simon first stage, and you might have a pre-specified threshold of activity. If you see fewer than that, you declare it inactive, and this has to be reported. One of the problems with research is that the anecdotal use of drugs may identify opportunities, but also opportunities where the drugs simply don't work. This needs to be reported so that people will not bark up this blind alley, and just as much as it needs to be reported if you have some evidence of activity. And I should say, the value of this is not where you see 3 patients out of 10 responding. The concept here is that science ought to identify situations where this is 8 out of 10, if we do it properly. It's missing those that causes me the greatest concern. And what would be a success for me? Success for me in this protocol would be that this process takes 12 months from go to O within the context of the framework, that it takes 3 months to get through ethics and about 7 to 12 months or so to get through the enrollment process, and you'll have answered this particular question. And that out of 10 protocols of this kind, one of them turns up a big hit because we suddenly identify an opportunity for a patient population which then proceeds to phase 2. Something like that. I think we need to explore that. This is part of what experimental medicine should be about, testing about whether this sort of model does work. Now, it's not about discovery. So I'm preoccupied with sequencing neochromosomes and discovering new patterns of mutations and doing animal studies, as many of my colleagues are. But this is not about that. So for somebody to go into this space and do whole genome sequencing, which will take 2 to 3 months to return the information to a patient with pancreatic cancer, is pointless. It's important that the genomics approach applied to the tissues that we have available in clinical practice, and I think this is one of the reasons why I think FFPE material is going to be important. So we've conducted a study, which I'll show you in a minute, using the Illumina TrueSeq panel, and we've applied that. Roughly the turnaround time for this is about 48 hours, which is very convenient. It has distinct patches. That's one of the reasons why this should not be a clinical diagnostic test at this point, but rather a research test, because the operating characteristics of these panels is far from perfect in an absolute commercial sense. And this just shows you some of the outcomes of this. So we conducted a multi-institutional study. This is 854 patients with various stages of disease, and you can see the disease subtypes here. The thing I want to draw your attention to is the average mutation rate for the 48 genes in this panel. And they vary from less than 1 in thyroid cancer and testicular cancer, and sadly it's also the case in sarcomas, up to 6 for patients with carcinomas of unknown primary. As you might expect, bowel cancers, which have a high mismatch repair deficient rate, have a high rate of mutations and so forth. But by no means everybody has mutations in this particular series. One of the things that I learned from doing this study over the past 3 years was that the detail is incredibly important, and you need to have the parallel development of both your molecular platform along with your trials. Now, fortunately, communities like Norway and Australia have already invested in much of this technology, so this forms a very important platform on which the clinical trials must be based. But I think turning that into a diagnostic as opposed to a discovery type of assay will be important. This shows you a little bit more detail about this is the cancer types and the types of mutations. You see P53 up the top, APC and colorectal cancer is very hot, but you see this very broad range of mutations across a range of cancer types, which just reinforces the point that saying you're going to study breast cancer and looking for targets without taking the opportunity to look at other cancer types is a missed opportunity in my view. So again, I think we should be sort of trying to do this in a histotype agnostic way. Nonetheless, using these 48 genes, which were highly selected, 63% had pathogenic variants according to our review process, 26% had therapeutic implications, but probably only fewer than 10% had opportunities for intervention. And I remind you about the BATTLE trial and the SAPPHIRE study. The rates of being able to translate for this group are not actually necessarily as high as you might think. It is important that we don't lose sight of other screening modalities. The screening test needs to be driven by the therapeutics, in my view, not the other way around. So I think designing the studies and doing the screening tests that would be relevant to those studies is actually an important thing. And that may not be all about genetics. There's been a lot of learnings. I won't go through this in detail, but this is a paper that was published last week in the British Journal of Cancer, which summarized many of the issues that we identified in the molecular pathology: sample quality control, inputs, sequencing platform, how to interpret the variants, how to filter them, and so forth, and how to report them are all really important details that need to be sorted out. So complex are some of these challenges that I think an important principle is centralization of this sort of process, because I think there are two challenges. One of them is to set up robust and rapid turnaround tests for our patients that is commensurate with their clinical outcomes. And I think the second is how you distribute that in a social sense through the healthcare system at numbers of sites. I think this is one of the arguments behind centralization at a limited number of institutions of this process. It may take within the next 5 to 10 years, this may sort of become part of standard care, but I think it's too early for that at this point. I touched upon this earlier, but I want to emphasize that I think it's a really important part of the design to have an engaged live adjudication process, probably by email and probably going to the level of everything from eligibility through adverse events, response and assessment of endpoints, not just the classical data safety and monitoring. But it does need to be independent of the investigator team. I think you need to still have some of the fundamental principles of having independence between your investigators and the review process. But equally, I believe without this live review, I think within 12 months of starting a protocol, you may find that you're doing so many amendments to keep pace with the generation of knowledge that it itself becomes rate limiting. So I think delegating to a group of respected individuals at a respected institution with a good and honorable track record of doing clinical research is an important way of developing a system which will satisfy the community's expectations of patient protections, at the same time accelerating the pace of development. And of course, I won't go into this in any detail, but that also applies to the Molecular Multidisciplinary Committee. I think interpreting genetic variation is a really complex process, and it depends upon circumstance. So what I mean by that is, if I have a patient who's dying and I have a previously undescribed variant which is highly likely to be pathogenic, my threshold for saying, let's put that patient onto a treatment will be completely different than if I go for a germline variant where I'm doing predictive testing in unaffected family members with all the consequent downstream impacts. There needs to be clinical context as well as molecular information that is brought together to the interpretation of this. And the other reason for doing that is because I think pace of knowledge generation is so great that unless you have a live expert committee reviewing these things and probably some review process at 12-month intervals, I think that it's not going to be possible to codify it in the classical appendix to a phase 3 or phase 2 process. So I think that's an important part of the future. Another question is how you prioritize sub-studies. So I think I just described some of the process that I think is very important. I think I'm driven personally by the sense of unmet need. If there isn't an unmet need in the cancer population, then this is pointless. You don't want to be treating people on a protocol when when there's good alternative treatments. So this is often for people at who don't have options to go into clinical trials, who run out of conventional therapies, but who are fit enough to go onto a trial. I think there needs to be a convincing, novel and testable hypothesis. Now that's more, you need to think about that. It means that if you have, for example, a patient with an amplification of CDK4, but there has been a study performed by Pfizer in well-differentiated liposarcoma, a disease in which CDK4 is amplified, and it's known to be ineffective, it's not a novel hypothesis. And it may be tested in another trial. CDK4 inhibitors in breast cancer have already been tested. So that some of the idea here has to be exploring opportunities where the questions haven't been addressed. That means you're trying to pick, in my view, populations where existing trials mechanisms have failed, not trying to compete with them. And there are some issues. It's important to avoid false negative results in the design of the trial. The worst thing you could do is say, this drug doesn't work in this disease or in this particular indication, and in fact it's because you have too heterogeneous a population, you've missed a signal of activity. That would be a very bad thing. So there may need to be an iterative model in this design. There has to be a suitable drug. This is not the space for testing new drugs whose toxicities are not understood. That's inevitably going to be a painstaking process of dose escalation, careful monitoring for toxicities. These have to be past phase 1, preferably non-toxic, repurposed drugs where we understand them. I'll give you an example. There is a disease called epithelioid hemangioendothelioma. Only a sarcoma specialist could love it. It has a translocation between the WWTR gene and a gene called TAZ. And TAZ, you've never heard of these genes, of course. TAZ is a homologue for a gene called YAP, which is a very important oncogene within the Hippo pathway, a newly emerging target for therapeutic development. TAZ is a homologue for YAP. There was a genes and development paper looking at compounds which inhibit YAP and identified a drug of a class of protoporphyrins. Protoporphyrins are in clinical use for photocoagulation. They're one of those labile compounds. You inject the laser and you get a reactive chemical and you basically photoablate proliferative retinopathy. So used for a completely benign purpose in the population. So an example of something that might be an interesting idea for a population which has no therapies, EHE, is to ask, could we treat those patients with protoporphyrin 9, or the chemical compound made by Novartis, which is used for photocoagulation, in a patient population with unmet need? I don't want to do that as one patient, as a one-off. I want to treat 10 patients, because I want to have an idea about whether there is something that's worthwhile pursuing in this. And I think this might be a perfect environment in which to do it. And again, I just make the point that that doesn't necessarily require a molecular hypothesis. It could be a clinical hypothesis or something derived from research, or many possible ways of getting into this. I said before, it's important that the question not already be addressed. If the question's been answered, I don't think we need to be doing the study. There are too many questions for creative minds and for active research laboratories to be wasting time asking questions again. And the other thing is, from a practical perspective, it has to be addressable in a reasonable time frame. This has to be successful. At the end of 4 years, I want to have both governments interested in this model so as to expand it to more broadly in the community, but I also want pharma to be persuaded. So that means, there are going to be many challenges along the way. Some of this is about the real politic of identifying the sweet spots that are not being addressed to make sure that we have. And that is not just a molecular question, that is a clinical question as much as it is, and a population. Is the population large enough? Is the response rate likely to be large enough, and so forth? And I think finally, although not critically, it's important that part of this premise is that if you get a hit, that you are going to go on to a phase 2. There are very few diseases where I think 10 patients is going to be enough to get registration for a drug. Actually, as it turns out, dermatofibrosarcoma protuberans and imatinib mesylate, that was approved on 8 patients in the B2225 study, but I think is unlikely to be the case going forward. So I do think it's important that component, the phase 2, go back to pharma for investment going forward. This is not something that I believe public health systems can invest in. So I think there needs to be a general premise that anything you put into this pipeline is something that is likely to be taken forward. So I thought I'd give you some examples about the modules that we're developing at the moment within this framework. The first 2 modules relate to PARP inhibition. There's a highly potent PARP inhibitor called BMN 673. We've had some discussions with BioMarin who have expressed interest. And the concept here is that if you have a mutation in the canonical target, BRCA1 or BRCA2, but outside of the indications in which these drugs have been tested, would you see the same sort of responses? So we enroll patients who don't have breast or have a sarcoma, for example. I happen to know, because we're doing a molecular screening program in sarcoma patients, that there is a very high rate of BRCA1 and BRCA2 mutations in the germline of sarcoma patients. They'll never get onto their own study, but we might be able to show that those patients may respond to PARP inhibition if they carry a BRCA1 or BRCA2 mutation. The second one is a different question. This complex within which BRCA1 and BRCA2 play a role has many other genes. FANCD2, FANCI, BRIP1, BARD1, PALB2 are just examples of these. If you have mutations in those genes and you have an ovarian cancer, would you similarly respond? That's an intelligent question that is worth asking, at least in a small population. It's also quite heterogeneous, which raises some interesting complexities about how you do this study. But in principle, anybody who has a PALB2 mutation could get treated with a PARP inhibitor on the basis of this study and it would be an intelligent, reasonable question to ask. The problem is that you need to be very careful about having a very heterogeneous group for a 10-patient signal-seeking study, because if it responds to PALB2 mutations predict response, but BARD1 mutations don't, then how will you pick that out of that mess? And the answer that we've come up with is that you need to integrate an iterative design. So this study could go on to a second phase of another 10 or 13 patients where one progressively enriches in hits in the biomarkers that will predict for a response. So this doesn't need to be regarded as a static process. Palbociclib, the CDK4 inhibitor from Pfizer, we thought about mutations in the RB pathway except for RB, which are not previously studied. These drugs have been applied in lots of trials. So this is a classic example where we want to look in areas that have not been previously explored to answer new questions. That would be the important thing here. Hedgehog pathway mutations, patched and smoothened, are much more common than we originally thought. I've seen a patient with a sarcoma with a patched mutation respond to the Novartis equivalent of this compound. I would like to know whether that just is a one-off or whether, generally speaking, non-Gorlin syndrome cancers with mutations in this pathway may respond, and so forth. One of the drivers for me about this is that I'm the head of a research institute with 217 staff and 12 laboratory heads. They're very clever, many of them, and many of them are promising to cure cancer and going into xenografts. I want to provide them with a technical apparatus, the experimental system, to be able to take those ideas into the clinic, and that's something only clinicians can do. Clinicians and clinician researchers and trialists need to think up some solutions that will help reduce the barriers to asking intelligent questions for people who are dying. Ultimately, the drivers behind this should not just be about research, but also about meeting unmet need. And I think having patients aboard is going to be an important theoretical principle behind this. So someone in my lab, Maya Kansara, who worked for some years on osteosarcoma, has this very elegant proof that this molecule IL-23 actually is required for tumor progression. And there is a compound, there are a variety of compounds which target this molecule, which are used for psoriasis and chronic inflammatory bowel diseases in the clinic, past phase 3, New England papers, been around for 5 years now. What if we took those drugs and asked whether there was some evidence that you could treat patients? Is this not worth testing 10 patients? What alternatives do they have? But then what mechanism do we need? What I've talked to you about today is a framework which we could think about, a creative framework for allowing those sorts of questions to be asked of patients who have no other better options. Thank you. Sorry. I've talked too long, I'm sorry.
No, thank you for an excellent lecture and also for pointing out possibilities for running clinical trials in new ways. I'm sure that since we have this beautiful mix of pharmaceutical industry, we have clinicians, we have research scientists. Hans-Petter Eikestad, yeah, good.
Thank you for a very nice talk and very kind of inspiring as a clinician to hear the kind of new ways of running clinical trials. My worry would be that with the trial design that you have, you would throw out too many drugs that might potentially be useful. I mean, you will from time, you will in some patients, obviously, you will in some groups find drugs that are very effective and work in 8 out of 10 patients, which is kind of those glory glorious stories that you want to read about, but some of these drugs are going to work in 3 out of 10 patients, and you
Yes.
might throw them out.
Yes.
So
So that's a really important point. So as I said, the worst thing you could do is rule out the possibility of future exploration. I think one important thing I didn't discuss here is that I think the leaders of a module need to be pharma, a clinician researcher, and a laboratory. And there should be companion biomarker for interpretation of the responses. So for example, if you had 3 out of 10 and you thought, well, is that enough to be going on with? If you had enough companion biology attached to those 3 patients, you might be able to then go to the next phase and then the iterative design and then select the biomarker for the 3 that did respond and see whether that would enrich progressively for the patients that did respond. I think this has to be associated with biological companion science for it to be valuable. I see this as being a mechanism which takes the last figure in a cancer research paper and makes it the last figure in a Nature Medicine paper. If you had 10 patients with a new hypothesis and you saw 3 out of 10 or 5 out of 10 responders, that would be a very dramatic and persuasive way to finish off a paper in that sort of quality. But your point's well made.
Ian Mills.
At what stage do you see scope for testing combinations with conventional therapy? Because obviously you put
Yes.
PARP inhibitors up there, and
Yes.
you hint at agents that will affect immune response, and so the obvious question is, in the end, do you get more efficacy by showing that your highly targeted, personalised therapy actually improves the survival in response to very widely accessible conventional therapy like radiation, for example? And who does that?
Well, I think it's really important not to be prescriptive about a module. I think, as I said, I could imagine situations where the idea comes out of not biomarker-driven space, but a compelling clinical rationale for a particular. Denosumab is a case in point. Denosumab works beautifully in giant cell tumour of bone. It's because of overexpression of rank ligand in giant cell tumours, but it's not biomarker-driven. So I think it could equally be the case that somebody makes an observation that people want to combine radiotherapy with PARP inhibitors. I think the one thing that you need, absolutely, is that there be a phase 1 data about how to combine the combination. This is not the circumstance where you could discover that. This is not a phase 1 study.
But in reality, they're all stress sensitizers, or a lot of them. The drugs you're putting up there are biologically predicting that you will enhance response to a cytotoxic as well.
Potentially, but those are just the drugs that I chose. There are more things in heaven and earth, I hope.
Gunnar.
Thanks a lot, David, for, as always, a very inspiring talk. My question was really along the lines of Ian's question, and that is, if your hypothesis indicates a combination of 2 drugs, where each separately has gone through phase 1, but the combination has not been adequately tested, would that be in scope for this approach?
I think probably not, because you take CDK4 and MDM2 antagonists. That would be a lovely thing to test for well-diff lipo, where you have co-amplification of both. But the problem is you don't know whether, when you combine the drug, that the normal cells, the marrow, for example, will tolerate the previous damage induced by chemotherapy. You might find that everybody falls apart. This is not the sort of framework for doing that. If, on the other hand, somebody had combined the drugs in breast cancer, so you knew how to use the combination, then it's completely reasonable to take that combination and the established RP2D into this sort of model.
But I would argue that even in this kind of design, or in this network of a few institutions
Yes.
cooperating on this, you might run into a hypothesis which is so attractive that you would invest in a phase 1 or a lead-in kind of test, or some way of approaching even the combination early. Because if you rule out that, sort of by principle, you rule out that opportunity, I think maybe some of the value here may be diminished.
Yes, no, I agree. I think the model you describe, where you set up a protocol where you establish an RP2D for a combination and then take it into this, fine. Yeah. I agree.
Giske Urshin.
Thank you for a very inspiring talk. I have a question about patient expectations and how they're changing over time. Because about 15 years ago, I was at the Comprehensive Cancer Center in the US, and there was a concern that the percentage of patients on clinical trials was so low. And it wasn't because of lack of protocols, and it wasn't because clinicians weren't trying to recruit them, but it was because the patients did not want to go on a clinical trial. They wanted the drug. And if they didn't get the drug, they would change to another center. So how do you see this? So let's say that you have these trials and you have more than 3 out of 10 respond, and then you want to push it into a phase 2 clinical trial. How do patients see this? They now know that this is a drug that they might find out that this is a drug where there has been some responders.
Yes.
Is it still possible to then recruit patients to clinical trials?
So I think you're right. People who have, I think that's a problem particularly of randomized studies where you have a 50-50 chance or something like that of getting the drug that you think might be working. This is not about randomization. It's not intending to make a comparison. In fact, it's not even trying to reach the standard of a single-arm phase 2 using conventional criteria that allow comparison with previous studies. It is just about essentially partnering with your patient to offer them more quickly an opportunity for getting access to what your expert, somebody like me who people get sent to see because I'm supposed to be an expert in my disease, I want to be able to translate that into, and my patients want me to be able to offer them something that is not just pulling it out of my back pocket and treating one patient and then forgetting about it, but in some structured way generating useful knowledge for the next patients that come down this path. I don't think there'll be a problem with enrollment of patients onto it. In fact, I would argue that the development of this sort of process improves the chance of avoiding patient cynicism and disappointment about paying $5,000 for a foundation medicine test to be told that their drugs don't exist in the first place for them to be treated. Because what we're saying is we will cover the cost of the test, but we will also give you, an increased chance of getting a drug, which is what they really want out of the test in the first place. I'm concerned that the 23andMe story, where 23andMe direct-to-consumer marketed a test which the FDA subsequently disavowed because they felt that the consumers were misled about their expectations of the test, could well be replicated in the somatic tumor testing if a smart lawyer realizes the patient really didn't consent to paying $5,000 for a test with no consequence, but rather for the purpose of treatment. And then I think this is why the things have to be put together. As to drug development, I can't promise that this will solve pharma's interest in developing a phase 2. That's something that's... The key thing about science is to ask a question which you can answer. I'm at this point just hoping that by doing this we will discover at least richer opportunities, more likely to succeed opportunities for phase 2 development. Whether pharma has the capacity or the interest to develop that, I think is going to take time. I suspect that over the next 4 years, if we do our job right and pharma see that there is some merit in this, pharma will start to co-invest in the infrastructure, which I think will be important for sustainability going forward. And hopefully part of that will be about having a sort of a structure for phase 2 development of the outputs of studies like this. But I think if we try to solve too many problems, we'll never get started. That's one of the characteristics of this time, right? Everybody's paralyzed because they can see far more problems than they can find solutions. I think just quarantining this to one testable hypothesis, which we can answer, is an important point.
Solbakk.
Two questions?
I liked your phrasing saying, genetically speaking, all diseases are rare diseases, and for this reason we need to change our research paradigms. But my question is, well, how do we safeguard rigid conceptions of scientific validity and clinical validity?
Yes.
And why is it then after necessary to do phase 2 trials and then eventually phase 3 trials? So wouldn't it be better to ask the question, would it be possible to develop research designs that deviates from the traditional randomized controlled trials, which is not economically affordable in terms of time and space, is completely waste of resources and patients, needs. Why a phase 2 after?
Why? There may be circumstances with orphan diseases where a regulator may accept a lower burden of evidence, and I mentioned dermatofibrosarcoma protuberans, where you have a good biological rationale and a clearly unambiguous response in a small set of patients. Regulators may well be driven by the Orphan Diseases Acts in the US and in Europe to approve things with a lower burden of evidence, particularly for a population which is unlikely to have a big impact upon the overall healthcare budget. But the reality is, I suspect that for more common diseases, it is scientifically desirable to make the evidence more robust. I don't see any way ultimately for more common diseases in actually asking the question whether the expensive new treatment is actually better than we currently provide, and in many cases it's not. I think getting the balance between that and not discriminating against rare disease where you couldn't possibly reach that threshold is important. So I think your question is really important, but it could be phrased another way as well. You said, how do you keep methodological rigor? One of the things about this protocol is that it's designed to be a semi-structured alternative to ad hoc use of drugs, which is occurring a lot across our communities. I think this speaks to the patient appetite and need in the community for options for therapy. This at least provides some way of recording the data in a somewhat systematic way and of evaluating beforehand whether everybody agrees that the idea is worthwhile. Clinicians are so variable.
Now I have 3 on the list: Steinar Omdal, Ola Myklebost, and Ragnhild Lothe.
Okay. To the last question about doing it necessary to do a phase 2 trial, I think it would be difficult to get a drug approved on phase 1 trial data or from the framework protocol only. We are now approving drugs from phase 2 trials. That was a challenge too, but we do that. So I think that's a bit too early to think in that direction. If I understand the framework protocol well enough, is what you intercalate a step. The phase 1 drug has been tested in phase 1.
Yes.
And you put in a small step. So 3 out of 10 patients, more than that, need to show some response.
Yes.
Right?
Yeah.
And so you talk about response. Is that objective response? Is it
Yes.
biomarker response?
Yes.
Because so what you do is reducing probably, it could at least be so, the numbers of phase 2 trials.
Yes.
So you reduce the numbers of maybe missing or obsolete phase 2 trials, which will never succeed.
Yes.
That's
That's
one
right.
thing. And the other thing is that these drugs, say phase 1 trial drugs, do you access those? That is a problem. I mean, some industry are interested in giving you the drug. They may have a trial, which is not the one you want to join. That has been a problem. If it's registered drugs, it's one thing. Then it's just reimbursement, although that's not trivial either. But access to drugs has been a problem for many.
Yeah, no, I think that's absolutely right. I do emphasize, I haven't shown the final slide because I talked too long, but this is essentially a partnership model in which public sector underwrites investment in genomic research, but puts that on the table to leverage further investment and interest from pharma. Pharma's going to be critical to this. They have to believe that this is useful. I don't think farmers are actually just as conservative as the medical profession in lots of ways, because they're driven by the regulators and the need to get reimbursed for their investments, and massive investments, and that's getting increasingly difficult. So I think this is probably going to be an acculturation process where we test it out at a set of institutions which are scientifically beyond reproach and which are conducting clinical research of the highest standards, just playing around with models of design that might bring them more into a partnership model. In Australia and probably in Norway, I think there's disengagement increasingly because of the high labour costs and the small populations that we represent in our countries. So I think from our point of view, or from their point of view, the thing that we can best provide is the high scientific value that we bring to the clinical research table. And I think thinking about how the government invests in that and sustainable models going forward will be something that we need to think about. And your first point, which is about...
Intercalating. So when you reduce the number of phase 2
Yes.
trials.
Yes.
Is that what you're thinking?
I think that we will what will happen now is that typically pharma go from biology and the results of phase 1 to designing their phase 2. And they typically pick the largest population, typically in a histotype-driven fashion for phase 2, because I think that's how they'll get their first... I think what we'll be offering will be a broader range of potential populations in which to screen patients so that they have a greater First of all, it means that you don't discover in 15 years, CML in 2000, GIST in 2002, DFSP in 2005. You discover these things earlier, and then the area under the curve, which is where they make their money, should be greater because they discover indications earlier in their drug patent life.
Ola?
So you touched briefly upon this reporting of negative results also, which of course is very important. And I wonder, is there a system where it's easy for an oncologist to find such information? And the stage 2 of the question is for all the ad hoc or off-label use, that probably is much larger than what you propose here. Should there be a requirement that this was reported in a more structured way so that the experience could be accumulated and shared?
Often drug companies will provide access to drugs on a compassionate program where they will provide access to the drug in return for information about the patient and ultimately about outcome. But it's not very organized. And yes, I think that would be an important thing from a social perspective. I think trying to regulate, there's an extremely heterogeneous group of clinicians, very diverse in their views and expertise. And I think being prescriptive about how they conduct medicine is probably politically a very challenging thing. I think clinicians are driven by their patients. If you can provide something that will help their patients, I think they'll get behind an idea like this. And that's the thing. We need to have some alternative to leaving them with a patient who is 25 years of age and is dying, but is ECOG naught. It's got to be better than guesswork.
So it could be a requirement for reimbursement that you report in a structured way.
That's a really important idea. So in Australia and probably in Norway, the regulators decide what they're going to reimburse. And one of the potential levers here, if we could do it right, I'm not sure how one would do it, is to get conditional approval for pharma for their drugs over a specified time where you collect the data, but where you also ask them to provide some of their drugs to the clinical research programs within our country for government-sponsored research. It could be that we use the regulatory carrot, which is what drives a lot of decision-making in pharma, to partly come up with a model. It's basically trying to develop the details around a collaborative model with pharma, which I think we need.
Last comment, Ragnhild.
Thank you very much. It's really tempting, of course, to be able to reduce the time from the idea and into the clinic, perhaps even avoiding going through the model systems. I just wonder, you mentioned this rare condition, dermatofibrosarcoma protuberans,
Yes.
which you had a response in 3 out of 8. So did you consider to the other 5, that subgroup, to investigate that together with other rare tumors that also originate from the same types of cells, like the neuroectodermal cells? That would be malignant peripheral nerve sheath tumors and perhaps even subgroups among melanomas that do not respond to the therapy they get.
Yes.
And if I'm not mistaken, I think all those 3 have amplifications on 17q. No obvious actionable gene, but perhaps there, for instance, there is a component, TK1, in the 5FU pathway. So what do you say about that type of approach?
Oh, it's fascinating. I didn't know the details about... in the case of the DFSP, 8 people all responded. On that B22-25 study, the JCO paper showed that every patient that was referred showed some evidence of response. And, you know, as clinicians, we treat patients, we see it. It's one of those cases where we got the right drug to the right patient through an exploratory low resistance trial mechanism. If it happened in 3, that's a good question. I think how we get scientists involved in designing the small modules is really important. We need to get the right bias specimens at the right time. Patients, in my experience, will, if they get an option for a treatment, will usually consent to having biopsies performed if it will help them and us understand what is happening to them. And I think this is the sort of study where I think there's going to be a lot of focus upon each individual patient as a very precious resource. So I think probably that's what an answer to your question. I think as you begin to explore other possibilities, you would have to engineer your companion science to anticipate some of the things that you say about whether the amplification on 17q is somehow correlated or not, and whether, you know, things of that sort. I have not answered your question, I know.
So thank you very much for this lecture. And on behalf of the program committee, Eirun has picked something for you to read on your long-distance air flights. And thank you for coming and enlightening us so much.
It is always a pleasure.
And now I just skipped to Norwegian. Vi lager en mikropause nå, bare for å fylle kopper og gå på toalettet, for nå vi så I godt klinisk utprøvningsmood, så nå går vi rett løs.
Toilet, I understand.